This is chapter 5 in section 1. Now we're going to get into biological molecules, the 4 major classes of biological molecules. A couple of things to get out of the way before we actually get into the molecules. The molecules are built from components. The components themselves are organic molecules, and they do have some uses, but the bigger molecules are strings of component molecules, are polymers, a term which will come up when we hit the first polymer in the starches. But to put 2 components together, you're going to build a bond between 2 hydroxyl groups, between 2 alcohol groups. And you're going to leave one of the oxygens behind as a bridge. Remember, oxygens makes a good bridge. That means that you've got to get an OH off of one side and an H off of the other side. So what comes out of this process is water. And you're making something. You're synthesizing a larger compound. That is dehydration synthesis. And not too surprisingly, if you want to break components apart from one another, which you do when you're digesting things, or you're breaking it down to reuse the bits and pieces of your own molecules, then you're going to put water back in with an H on one side and an OH on the other side. And so you get a piece off. Water goes in, piece comes off, whether you're doing it with proteins, pulling amino acids off, whether you're doing it with a string of carbohydrates, whether you're doing it with lipids. Those are all things that'll come up in the process of this chapter. Now, this often happens with biological terms. Notice I didn't write that. I'm assuming you're writing down how dehydration synthesis works. Because that's what you gotta do. Very often you'll get pairs of terms. And unless you took a couple of years of Latin, which I did many years ago, and it's been very useful, very often one of the 2 terms is easier to remember a lot of times because it's more Englishy than the other term. And then the way I learned it is I remember the 2 terms. The one that's Englishy, I can remember how that one works, and then the other one is just, oh, that's the opposite. You're making something. Water comes out. Dehydration synthesis. What's the opposite? Hydrolysis. Water has to go in, you're breaking something apart. Now, if you know Latin, this is water breaking, literally, water breaking. And so that's what's happening. You're putting part of the water on one side and part of the water on the other side. You're breaking the water apart to do it. So those are standard assembly, disassembly reactions in not just organic molecules, but a lot of other molecules as well. So as far as the groups are concerned, we start with the carbohydrates. Carbohydrates, carbon and water. The simplest of the carbohydrates, that's the proportion of the atoms. For every carbon, you have two H's and an O. The smallest ones are sugars, glucose, C6H12O6. You've got, C4H8O4. You got a whole bunch of different sugars. You've got dextrose, which is glucose rearranged. It's an isomer of glucose. Same, it's C6H12O6, but they're put together a little bit differently. And as long as they're small molecules, it could be a single sugar molecule, it could be a couple put together like the overhead had, it could be 3. You can have what they used to call saccharides. They still do monosaccharides, disaccharides, trisaccharides. You don't tend to get medium-sized carbohydrates. You tend to get short strings and really, really long strings. Glycans could be sometimes medium, but even they tend to be fairly short strings. Oops, let me. Yeah, I'll get it on here eventually. There's rings. The individual sugar molecules are ringed molecules with carbons at the corners except for one corner, which is an oxygen bridge. That is the spot where the ring occasionally opens up and forms a string. It tends to be a temporary arrangement, but I'm going to keep saying this. You have to keep in mind on the atomic level, things are shaking and moving and bouncing off of one another. There's a lot of activity, a lot of space, but also a lot of stuff. And molecules shake, and sometimes at a weak point, they pop apart and then they pop back together. We talked about how water, a small proportion of any water molecules in water have popped apart and will reform eventually. But this is your typical ring with the oxygen bridge. This is the opened ring. And in us, when those rings are open, they're much more chemically reactive than when they're closed. And closed sugars are pretty chemically reactive. There's some evidence that a lifetime of being exposed to these occasional molecules reacting with things in your system you don't really want them to react to may be one of the contributing factors to aging. People that are diabetic from an early age, if you look at their symptoms as they get older, they in many ways tend to age faster. So that's a suggestive piece of evidence that there's a whole bunch of things that cause aging. This may be one of the things. Free radicals of oxygen or another thing, genetic timing, epigenetics, all sorts of things contribute, but this is one of them. Sugars are generally used for energy. They do provide, differentiation of markers on cells. When you take in cane sugar, the glucose is used directly for energy for the most part. The fructose, which is the other half of sucrose, the fructose actually is opened up and integrated into lipid molecules more than it's used directly for energy. But lipids can be used for energy too. So the little bits, if you're looking for things that they do, you can get away with going, they're pretty much an energy supply. Now, if you remember from the first semester, respiration is a process where you take energy from a fuel molecule and you transfer it into a more useful form, usually ATP, but as we'll learn as we go through the semester, there's other places that you can move it as well. And when you're learning about aerobic respiration and anaerobic respiration, the basic fuel molecule you start with is glucose. As we also talked about, you can feed, as long as you've got carbon and oxygen and hydrogen, you can feed a whole bunch of other things in small bits into the respiratory pathways and pull them apart for energy as well. But sugars, upfront, primary fuel for respiration. The origination point for an awful lot of sugars is photosynthesis, whether it's in plants or protists or archaea or Monera. There's a whole bunch of different groups that have photosynthetic species in them. But you're taking light energy and essentially converting it to bond energy. It's an indirect conversion. You don't directly make sugar bonds, carbon to carbon bonds with the bond with the light energy. You go through ATP and on the way. We'll talk about that later. But this is where you're tracing most of the sugars back to. Most of our fuel can be traced back eventually to sunlight. Starches are long strings of sugar molecules. Whoops, sorry. Long strings of repeating components. They don't have to be identical components, but they have to be the same sort. So in this case, they could be different sugar molecules, but as long as they're sugar molecules, you have a polymer. And starches are polymers, long strings that can be bound together in different ways. They can be very simple strings. They can be just somewhat simply branched strings. They can be strings with a lot of cross connections. And that makes them different. The different bond types are called linkages. A whole bunch of stuff of connections like this are alpha linkages. A bunch of connections that go like this are beta connections. It turns out that the alpha connections are much more easily broken later than the beta connections. And so why? Well, the plants use alpha connections to store sugars that they're planning on using later. They want those connections to be breakable, because they're storing starches in their roots, or storing starches in various places where later they're going to use it for what plants use sugars for. We'll talk about that in a second. In beta connections, they want those starches to be very stable, to be long-serving starches. Is it beta connections, hard to break. And it really is the difference between what we consider digestible starches and indigestible starches. Is that what our systems can actually break down and get the sugars out of, or what we consider to be plant fiber and undigestible. It's not easy to digest plant fiber. There's very few animals that can do it with their own internal chemistry. Almost every animal that eats plant fibers and digests it are using little buddies inside them, mostly bacteria, that can break the beta linkages. There's a tremendous amount of potential energy in there if you can get the sugars apart, but it tends to take a pretty dedicated, specific type of digestive system. Plants do have energy requirements. They do need the sugars, once they've made them, they may use them later to do things that require energy. But a plant is made largely out of starches. So the fibers that you got to crunch through to eat a piece of celery are the fibers that you can't really crunch through in order to eat a piece of wood. There's some proteins that add to the reinforcement structure of wood. That's what plants use them for. You don't tend to find starches being used for structure in animals. In funguses, you use kind of a weird offshoot of a protein, sugar, lipid kind of mixture that I'm drawing a blank on the term for right now, that are also used for structure. Okay, what I'm going to try to do is break up the different videos for the different types of materials, which are going to make them fairly different lengths. So this should be it for carbs.